Usually, human development is something we just expect to be entirely visible. Like, we're so used to seeing those incredible time-lapse videos where you watch, a single cell divide and then the embryo forms.
Right, yeah.
You see the little flutter of a heartbeat and eventually, boom, there is a fully formed infant. It kind of creates this illusion for you, listening at home, that we have a complete, uninterrupted map of how we're built.
Yeah, it's a completely understandable assumption to make. I mean, we map the human genome, we have all this advanced MRI imaging, so it really feels like every single stage of human biology should be perfectly documented by now.
Right, but then you step into the world of neurodevelopment, and specifically I'm talking about the period right around birth, and suddenly the camera just cuts out.
It really does.
We are looking at a developmental landscape that is just a total black box.
Right.
So today, our mission is to crack that box wide open. We are taking a deep dive into a massive and frankly kind of mind-blowing new study published in Nature Communications.
It's incredible work.
It really is. The researchers utilized transcriptomic and proteomic data from 336 brain samples to figure out exactly how our brains wire themselves right before we are born.
Yeah, and it is just a phenomenal data set because the perinatal period, which spans from late pregnancy to early infancy, is, well, you could argue it's the most critical chapter of mammalian development.
For sure.
But because of, completely valid ethical and logistical barriers, studying intact human brain tissue dynamically during this exact window is it's nearly impossible.
Yeah. Okay, let's unpack this because the stakes here are just enormous. During this specific perinatal window, your brain literally triples in size.
Triples, yeah.
It quadruples its cortical surface area. And we know the brain goes into this phase as just a basic biological framework, and it comes out ready to process a complex, chaotic world. But how it transitions from raw materials to a functioning network has been a total mystery.
Exactly.
So to get this multi-scale map, scientists had to turn to an incredibly unlikely model, not a new AI, not a super resolution MRI, but the Bama miniature pig.
Right, and that is the real breakthrough here. And I know it sounds surprising. Usually when we read about biological studies, the default model is the laboratory mouse.
Always mice.
Always mice. But there is a fundamental evolutionary gap when you try to study perinatal brain development in mice. It really comes down to two things: architectural complexity and developmental time.
Wait, I always just assumed a mammalian brain was a mammalian brain, just scaled up or down. Why can't a mouse show us what our brains are doing?
Because the architecture is completely different. Mice possess what neurobiologists call a lissencephalic brain.
Lissencephalic.
Yeah, lissen meaning smooth. It completely lacks the deep folds and wrinkles, the sulci and gyri, if we want to be technical, that really characterize a human brain. Humans have a gyrencephalic brain.
Okay, so it's all folded up.
Exactly. Those folds are just an evolutionary workaround. They allow us to pack a massive amount of cortical surface area into a relatively small skull, so, we can actually fit through a birth canal.
Oh, wow. So a smooth brain literally doesn't have the real estate or the structural complexity to mirror our own. If I'm picturing this, it's a mouse brain is like a single-story studio apartment.
I like that, yeah.
And our brain is a massive skyscraper. Yeah. You can't learn how to engineer a skyscraper by watching someone nail together a tiny cabin. The physics, the internal wiring, the structural loads, it's an entirely different league of engineering.
The skyscraper analogy works really well for the scale, though we do have to remember biology is much more dynamic than, steel and concrete.
Right.
It's a building that is actively remodeling itself while the scaffolding is still going up. But the second, perhaps even bigger issue with mice is the timeline.
The timeline.
Yeah. A mouse pregnancy lasts roughly 21 days. Their development is so rapidly compressed that they entirely skip this prolonged, complex perinatal transition period that we humans experience.
Wow, 21 days. Which brings us to the Bama miniature pig.
Yeah.
Why are they the perfect proxy for us?
Well, they check every critical box. They have deeply folded gyrencephalic brains. They have a very similar brain-to-body weight ratio to human infants. But most importantly, they experience a prolonged perinatal brain expansion that mirrors the human timeline in a way a mouse never could.
But how do we know the timelines actually match? I mean, a pig is still a pig. You can't just assume a certain day in pig development perfectly mirrors a human's third trimester, right?
You're absolutely right to question that. And the researchers didn't just guess. They used a highly sophisticated algorithm called RAPTOR.
RAPTOR? That sounds like something straight out of Jurassic Park.
Right. It stands for Real Age Prediction from Transcriptome Staging on Reference. Essentially, it's biological time travel.
Oh, that's cool.
Yeah, RAPTOR is a cross-species age mapping algorithm. It takes the massive data sets of gene expression from the developing pig brain and systematically aligns them with existing fragmented data sets we have of human development.
So it's looking for overlaps.
Exactly. It looks for homologous developmental anchors points, where the global gene expression patterns match up perfectly across species.
So it's mathematically proving the timeline alignment.
Yes. And the alignment is stunning. They proved that the pig's embryonic days, 76 to 104, we'll call that E76 to E104 maps perfectly to human post-conception days, 183 to 257.
Okay, let me pause and synthesize this for you listening, because this is really the foundation of the whole deep dive. We are trying to illuminate this black box where the brain triples in size. Point 1, mice develop too fast and have smooth brains, so they can't show us how this works. Point 2, through the Raptor algorithm, we now have mathematical proof that looking at a folded Bama pig brain at E76 is essentially looking directly into the human late third trimester.
That is the core premise, beautifully stated.
Yeah.
And once they established that proxy, they didn't just take a surface-level look. The scale of this methodology is staggering. They analyzed 336 separate samples.
Right. 336 samples across 8 distinct developmental stages. And they didn't just, grind up the whole brain into a smoothie to see what was inside.
No.
They painstakingly dissected 7 different brain regions, like the prefrontal cortex, the hippocampus, the cerebellum, the striatum. But what really caught my eye is what they were measuring in those samples. It wasn't just genetics.
No, they conducted what is known as a multi-omics study.
Multi-omics?
Yes. At every single region across every stage of time, they measured 2 distinct biological layers, the transcriptome and the proteome.
Okay, for those of us who haven't taken cellular biology in a while, let me try to translate this. Let me know if this mnemonic works.
Go for it.
The transcriptome is all the RNA. I like to think of RNA as the architectural blueprints or the recipe. RNA equals recipe.
Yes, perfect.
It's the information telling the cell what to do. Then the proteome represents all the proteins. Those are the actual bricks or the baked pastry. Protein equals pastry. It's the physical material that actively executes functions and builds the cell structure.
The recipe and pastry mnemonic is highly accurate. You absolutely need the RNA to know what to build, but the proteins do the actual heavy lifting. What's fascinating here is what happens when you measure both of those simultaneously across time.
Right, because usually we just look at one or the other.
Exactly. The researchers found that while over 80% of the proteins match their RNA recipes, meaning the brain is generally following the instructions, there is a significant temporal lag.
A delay.
Yes, a delay. The brain doesn't just print the RNA recipe and instantly bake the protein pastry. Transcribing RNA is relatively fast, but translating that RNA into a complex, three-dimensionally folded protein that takes significant cellular resources, transport mechanisms, and energy.
So it takes time.
So there is a delay, yes.
And reading through the data, I noticed this delay isn't happening uniformly across the brain, right? Different neighborhoods seem to be building themselves at entirely different speeds.
That brings in a really crucial concept they coined developmental velocity.
Developmental velocity.
Yeah. If you look at the cerebral cortex, those outer, highly folded, evolutionarily newer layers of the brain, it develops as a highly synchronized team. The prefrontal cortex and the motor cortex, for example, are moving at a very similar velocity.
And you're pacing each other.
Right. But when you look at the subcortical regions, the deeper, older parts of the brain, like the thalamus or the striatum, it is highly heterogeneous. They each have their own biological speed limits.
Wait, let me slow down and make sure I've got this. If the brain is tripling in size, and we have these blueprints and proteins lagging behind each other, plus different neighborhoods operating on totally different biological speed limits, I mean, that sounds like a recipe for absolute chaos. Is it just chaotic construction, or is there a master schedule governing all of this?
That was the defining question of the research. If it were chaotic, the brain would simply never function. There has to be a master schedule.
Right.
And when they mapped all of this complex omics data chronologically, they actually found the master schedule. They discovered a massive pan-regional inflection point.
Okay, let's do our second mini recap here to lock this in. 1, this study looks at both the RNA blueprints, the recipes, and the protein building blocks, the pastries. 2, the cortex develops together as a team, while deeper brain regions are much more varied. And 3, there's a biological speed limit that varies by region. But now you're saying they found a pivot point in the schedule. Where does it happen?
It happens between embryonic day 94 and day 104. When you plot the developmental changes on a graph, the data literally creates a deep V-shaped trajectory. E104 is this magical turning point.
E104, what is actually changing direction at the bottom of that V?
It's a complete shift in biological priority.
Yeah.
Before E104, the brain's developmental velocity is heavily skewed toward neurogenesis and axonogenesis.
Meaning?
Essentially, it's just trying to manufacture billions of raw cells and run the initial bare electrical wires, the axons between them, to establish a really crude framework.
Got it.
But at E104, the entire system shifts gears. It stops focusing on mass-producing cells and abruptly pivots to synaptogenesis and myelination.
So it shifts from just throwing parts into the skull to actually wiring the network together for function.
Exactly.
Let me go back to my house analogy for a second. Before E104, the electricians are just pulling thousands of miles of raw copper wire through the walls. It's chaotic. There's no power running yet. It's just raw connections.
Yeah.
But at E104, they suddenly start wrapping every single one of those copper wires in thick rubber insulation so that when you finally flip the switch, the whole house doesn't short circuit.
That rubber insulation is exactly what myelin does. Myelination doesn't just prevent short circuits, though. It dramatically increases the speed at which electrical signals can travel down the axon.
Oh, so it optimizes it, too.
Highly optimizes it.
Yeah.
And the data perfectly supports your analogy. The researchers observed a massive brain-wide surge in a very specific protein called MBP, or myelin basic protein, and it peaks right at E104.
So the whole brain collectively decides at E104 to start insulating the wires.
Well, almost the whole brain. Biology
Okay.
always has an exception.
Of course it does.
There's one fascinating anomaly in the data, and that is the cerebellum.
Ah, the cerebellum, the little brain tucked at the back of the skull near the brainstem.
Right. The researchers found that the cerebellum operates on the absolute shortest developmental time span of any region they studied. It matures precociously.
Precociously.
Yeah, while the rest of the brain is still busy running those raw copper wires in the early perinatal period, the cerebellum's cellular construction phase, specifically its gliogenesis, has already peaked. By the time it hits the perinatal period, the cerebellum is already fully focused on refining its circuits.
It's the overachieving early adopter.
Exactly.
It finishes its basic construction while the cortex is still figuring out where the walls go, and it just sits there optimizing its network. But why would the cerebellum rush its development like that?
Well, think about evolutionary survival. It makes perfect sense. The cerebellum is the primary control center for motor coordination, balance, and vital survival reflexes.
Oh, like breathing and swallowing.
Right. An animal or a human infant absolutely needs those essential motor circuits online the second they leave the womb. You can afford to have your higher-level thinking cortex mature slowly over years, but if you can't coordinate your breathing or your basic balance immediately at birth, you just won't survive.
That is incredible. Okay, recap number 3 for you listening. Point 1, E104 is the magic pivot point where the brain starts insulating its wires. That's myelination. And point 2, the cerebellum is the overachiever that finishes its basic construction first to keep us alive. But as I was reviewing the study, there was a dynamic that really puzzled me.
What was that?
The researchers talk about a cup-shaped model of heterogeneity. What exactly is taking the shape of a cup here?
Right, so heterogeneity in this context refers to how molecularly different one brain region is from another. You might intuitively assume that as the brain grows and specializes, the prefrontal cortex and the hippocampus would become more and more distinct from each other.
Yeah, you'd think they drift apart.
But the data shows the exact opposite.
They become more alike.
They do. The differences between the regions actually peak quite early, around embryonic day 85. At E85, the regions look wildly different on a transcriptomic level. But as they approach the birth window, those interregional differences drastically fall.
Oh, I see.
Plotted on a graph, this heterogeneity forms the shape of a cup, reaching its absolute lowest point, its highest level of synchronization, right after birth.
Okay, so it's like all the different neighborhoods in a city are building completely different styles of houses. Victorians, modern, ranches. But right before move-in day, they all synchronize and decide to paint them the exact same color.
That's a great way to visualize it.
But here's the part that confused me. The RNA differences. The recipes synchronize and drop right around birth, but the protein differences, the actual physical pastry, don't peak until much later, around E109. If the blueprints synchronize, why are the building blocks still doing their own wild, heterogeneous thing days later? Who is managing this delay?
This raises such an important question, and it's a real testament to how deep this multi-omics approach goes. The researchers dug into the molecular managers governing this exact delay.
Who are the managers?
They identified two major classes of regulators: transcription factors, or TFs, and RNA binding proteins, or RBPs.
Here's where it gets really interesting. How do these two differ?
Well, they operate at completely different stages of the process. Transcription factors, the TFs, act directly on the DNA to regulate the RNA blueprints. The studies show that TFs are bidirectional.
Meaning they go both ways.
Yes, they act as both gas pedals and brakes. They can upregulate the reading of a recipe, or they can completely repress it. They are basically the strict traffic cops of the genome, carefully directing the flow of blueprints.
Okay, so TFs are the traffic cops managing the RNA. What about the RBPs?
RNA binding proteins act on the translation step, the actual process of turning that RNA recipe into the functional protein pastry. And unlike the bidirectional traffic cops, the data showed that RBPs are heavily biased toward positive regulation.
Positive regulation?
Yeah, over 60% of them are actively pushing translation forward. If TFs are the strict traffic cops, RBCs are the enthusiastic project managers on the construction site, constantly yelling at the cellular machinery to build faster, build more.
So the traffic cops are tightly organizing and synchronizing the blueprints, causing that drop in RNA heterogeneity. But downstream, you have these enthusiastic project managers pushing the actual physical protein construction forward at different rates across different regions, which creates that massive delayed peak in protein heterogeneity at E109.
Precisely.
That mechanism is brilliant. Okay, let's do our fourth recap. 1, brain regions look most different from each other right before birth, then they synchronize into a cup shape. 2, proteins lag behind RNA. And 3, RBPs act as the molecular accelerators to get those proteins built.
It is a brilliantly complex system, but it also highlights a profound vulnerability. And this brings us to the ultimate point of the entire paper.
Yes, because as fascinating as molecular traffic cops and pig cerebellums are, you listening are probably wondering, why should I care about this? How does understanding a temporary protein lag in a pig brain translate to human health?
It matters intensely because of what happens when this perfectly orchestrated, highly vulnerable construction schedule is interrupted.
What happens?
The researchers took known human genetic risk factors for major neurodevelopmental disorders, conditions like autism spectrum disorder, developmental delay, and lissencephaly. And they mapped those human genes directly onto the pig's developmental timeline.
Because they have Raptor, they don't just know what the genes are, they finally know exactly when they activate in the timeline.
Exactly. And the results they found completely reframe how we view these conditions. They found that during the early perinatal window, that E76 to E94 period before the big inflection point, the genetic network of the brain is highly disorganized. It is fluid. But right as the brain hits that E104 inflection point and moves toward the days just after birth, the entire genetic network undergoes a massive reorganization. It locks into an adult-like, highly correlated pattern.
The concrete set.
The concrete sets. And here's the critical discovery. They found that the specific risk genes for autism and developmental delay genes like SYNGAP1 and JBRB3 don't just randomly float in the background.
What are they doing?
During this exact transitional window, they act as highly connected network hubs.
Network hubs. So if we picture the developing brain's genetic network like an airline map, these genes aren't some tiny regional airport in the middle of nowhere that only handles 2 flights a day.
No, not at all.
They are JFK, they are London Heathrow. Vast amounts of biological information and developmental routing flow directly through them.
That's a great way to conceptualize it. SYNGAP1, for instance, is crucial for regulating synaptic plasticity, while GBRB3 is vital for inhibitory neurotransmission. They are structural pillars of the network. If we connect this to the bigger picture, it means that if something disrupts these specific hub genes during this incredibly brief, fragile perinatal window.
Like what kind of disruption?
Whether that disruption is a genetic mutation, an extreme environmental stressor, or premature birth, it doesn't just cause a localized temporary problem.
Because it happens right at the V-shaped inflection point.
Right
as the network is finalizing its adult shape.
Yes. A disruption to a hub during that exact window alters the routing of the entire adult brain network permanently. The perinatal period isn't just a phase of rapid physical growth. It is the crucible where the lifelong functional integrity of the mind is forged.
Wow. Let's pull all of this together for our final recap. We started out looking at a black box, the inaccessible period right around human birth where the brain explodes in size and complexity. Mice couldn't help us. But by using the gyrencephalic Bama pig and the Raptor algorithm, we proved that E76 to E104 gives us a direct window into human development. We discovered that the brain operates on a master schedule, hitting a massive inflection point at E104 where it stops just building cells and starts heavily insulating its network with myelin.
That's the V-shape.
Right. We learned about the traffic cops and project managers coordinating the blueprints and the bricks. And most importantly, we learned that this exact window is when the central network hubs for major neurodevelopmental disorders lock into place.
You've summarized it perfectly. Knowledge is most valuable when it is understood and applied. For decades, neurodevelopmental conditions have been somewhat static mysteries.
Yeah, we just didn't know the timing.
But now we have a dynamic spatiotemporal map. We have the recipe, we have the building blocks, and we know the exact temporal window when the network solidifies. We finally understand how the scaffolding of our minds is actually erected.
So what does this all mean for the future? I want to leave you, listening to this right now, with a thought to mull over. We established that diagnosing and treating neurodevelopmental issues is incredibly murky. It's not a clean X-ray of a broken bone.
No, it's not.
But now we know about the E104 inflection point. We know the exact window mapping right to our late third trimester where the network concrete sets. If we know exactly when the network solidifies and we know exactly which hub genes, like SYNGAP1, are driving it, could future medicine utilize this highly specific temporal window to deliver targeted software patches?
Oh, wow.
Could we eventually develop therapies that correct these genetic network hubs for conditions like autism or developmental delay dynamically before the concrete of the adult brain fully sets?
It is an incredibly provocative question. The biology is profoundly complex, obviously, but for the very first time, we actually hold the map that could guide us to those kinds of interventions.
We finally have the blueprints to the skyscraper. Thank you for joining us on this deep dive. Keep questioning the world around you, and we'll catch you next time.